Hello, everybody, and welcome to the Diatom Web Academy. This webinar series is recorded and free of cost, and it will have an introduction of key topics on diatom biology and applications of diatoms to environmental science. I want to introduce our webinar speaker today, Dr. Sarah Spaulding. She is an ecologist at the US Geological Survey. She is a research scientist at the Institute of Arctic and Alpine Research and affiliate faculty at the University of Colorado Boulder. She is our fearless leader, creator, and webmaster of Diatoms of North America website. And I want to include the quote that she has on her profile in the Diatoms of North America website. The truth about diatoms should be accessible to everyone in cyberspace and beyond. So please take it away, Sarah.
Thanks, Sylvia. And I wrote that maybe 10 years ago, and here it is. We're finally become a reality that we can connect with people across the world working on diatoms. So I'm really pleased to start off what we're calling the Diatom Web Academy with Sylvia and the rest of my collaborators who are all part of the Diatom Taxonomic Certification Committee. I wanted to make sure everyone make sure that you have the chat window open. If you have any technical issues, you can check in with Sylvia and we'll try to solve them. And if she has any points of clarification, Sylvia will be looking at the chat window and can relay those things to me or just say something. And then at the end of the presentation, we'll have Sylvia read off your questions, comments, concerns, feedback, and have a chance for discussion. So with that, we'll get started. So I want to actually, first I'll start off with the way I'm organizing the slides. On the left-hand side, I've got a background, and so that'll be, if you need a little more information, you can consult that stuff over there, and I'll be talking mostly about what's on the right-hand side of the slide. And so what I will start off with is this problem, and that is the Diatoms of North America project, which we call DONA for short, is establishing a collaborative source of taxonomic and ecological information on diatoms. And it has come to serve as the resource for the Level 1 Taxonomic Certification Program, and it's also heavily used by practitioners around the world. But practitioners, people working on diatoms, are asking for more support in their work and research, including more support for training and certification. So our group is trying out this forum as a means to do that. Our solution is we're offering this webinar series to introduce key topics concerning diatom biology and application of diatoms to environmental science. We will have presentations that are by our group, but we'd also like to invite in people from other avenues, from academia, from different countries, and we intend that the sessions will allow for time for feedback and discussion, that Sylvia and I, as being finding ourselves working for federal agencies, find that we need to translate. we need to be able to be in the role to talk about the way diatom biologists think and talk, and the way managers think and talk. And so we're seeing the value of doing that, and we'd like to broaden that conversation and engage all of you in talking about the issues and how we go about solving them. So who are we as part of the Taxonomic Certification Committee? The current members are from agencies, academia, consulting, graduate students. We have been working together for over a year now to develop the first level certification program. And we're working further to clarify higher levels of certification and just what kinds of training and support we need to do that in order to have a program that supports taxonomic consistency, transparency in data, and accessibility to the people who need the information. So just as a introduction for this first webinar, we want to reach out and let you folks know what we think you want, but we want to confirm that. We have over on the left, if you go to that URL, we have, it's a place where you can give us feedback on the proposed series of webinars. And these webinars will be recorded. This one will be recorded, and we hope to have it up later in the day on Diatoms of North America. And we have a variety of topics. Some of the proposed webinars are very much assessment and management related, like a brief guide to algal contracting for managers, designing a study with a clear purpose. Can I reach my goal with diatoms? What data do I need? Another potential webinar is how are diatoms used in assessment? How to set clear goals, and why use any particular group, whether it's diatoms or soft algae or molecular sequences. What can I do with diatom data? When should I use indicator taxa, thresholds, multimetric indices, or O/E models? Diatom multimetric indices, diatom physiology and ecology. What is the fundamental knowledge underlying the use of diatoms in assessment? Those are some of the ones on the series that you can respond to if you go to that, that link. And some of the other proposed webinars are maybe more in depth for analysts or people who, students who want to look at a paleontological question, reconstructing the past. What's the process? How do I go about obtaining sediment cores from lakes? What are the field and equipment needs? What is a chronology and how much does it cost? Molecular approaches to assessment. There's been a lot of changes and a lot of changes going on. Where are we now with collecting, preserving, and extracting DNA from samples? The genus X, morphology, ecology, and distribution. We can dive into the genera that are informative about water quality, or just that genera that people want to know more about. And there are people who are experts in particular genera across the world, and we'd like to invite them in to give a seminar. So let us know what you're thinking about. Light microscopy and imaging diatoms. We might talk about tips, and troubleshooting problems with optimizing light, how to calibrate between analog and digital measurements, photography. So please take the opportunity to let us know about which of these topics are appealing to you, or feel free to propose other topics that you would like to hear about that would be helpful to you. And we will work at setting up what will be the sort of whole series of webinars for this year. So we want to know who you are, the audience. Today, I've directed this talk at people who work in water quality, and we're thinking that we want to reach out to managers, tribes, state agencies, federal agencies, consultants, ecologists who are charged with working with algal data. And from the previous slide, some of these webinars will be for assessment needs, and some of the webinars we'd like to have directed at analysts, students, ecologists, broader audiences. So this really gets back, I mentioned this information gap that we need to be able to talk and translate. Science, we scientists can say stuff about diatom biology, and managers say stuff about assessment, and often the understanding is garbled or just lost. And we're here to translate. We want to be the people that you feel like you can connect to. There are too many urgent environmental problems right now for anyone to waste their time or your precious time or the few resources that are available. We have the ability now, through connecting through forums like this. We want people to do good work. We want to do good work. We want you to do good work. We want to make that possible. So, all right, let's go. Why are diatoms used as indicators of environmental condition? Some of the things that people figured out really early in the 20th century was that diatoms have tolerances and optima to certain environmental conditions, and that the traits that diatoms have reflect the way that they acquire resources like light or nutrients, and their resistance to disturbance. And so I'm going to give a little bit of an introduction to diatoms and the way that knowing something about them and knowing something about their preferences can be really useful in assessment. So to say something has a narrow tolerance is another way of saying that some diatoms are picky. And some diatoms are picky about the pH of the water that they live in. For example, many of the species in the genus Eunotia like to live in acidic water and generally in high in dissolved organic carbon. And I'm going to talk first a couple examples that come from lake sediments. This text is from a publication that constructed 12,600 year record of the acidification of a lake in Sweden. And this is really landmark publication. The diatoms showed the history of the lake and their pickiness about pH over that over 10,000 year period. And some of this work was really fundamental in determining the acidification of lakes, particularly in Europe, from sulfate deposition. And what Renberg did in this 12,000-year plot was characterize the diatoms by their pH pickiness. So this looks pretty complicated at first. There's a lot of black. And can you see my arrow? Yeah, okay, good. Over here is the 0. This would be the present time, so when this study was done. And time runs backwards as we go over to this side, 350. So this is really depth. Oops. Rats. Okay. Depth in the sediment. And if we go down to the very bottom here, the bottom sediment is worth over 12,000 years. And then we can see 7,000 years ago, 23,000 years ago, 1900 AD, and all the way up to at this time when this was done, just cored in 1986. And what this record shows is the relative frequency of diatoms and their preferences. And this big, I'll be able to start at this big one in the middle, indifferent. The diatoms plotted here, their frequency, they didn't care too much about the pH. Diatoms that like really alkaline water, the alkali-ophilous, alkali-loving, decreased over time from 12,000 years to the present. The acidophilous diatoms, the ones that like acid, went from being relatively low and increased over time with some bumps. And the acidobiontic, which the ones that really have to have acid water, here is the key part of this whole graph, that in this plot, this increase right here in recent time was a human-caused acidification. Now so here we've got a giant graph, and a little tiny part that was really part of the crucial story. And so it really, I think, kind of emphasizes understanding the record and what the data is showing. So that was an example from lakes and reconstructing the pH record in a lake over a long period of time. We could talk about diatom pickiness, about nutrient concentrations like nitrogen and phosphorus of the water that they live in. And again, I'm going to talk about a lake sediment record. In this sediment record, we've flipped the axes a little bit to the way most sediment records are reported, which is the 0, which this was about the year 2000, going back to 20 centimeters of depth in the record. And the age of the sediments, this dotted line, represents 1960. And what this plot shows that if we start over here at 1960, things started to happen in this lake in 1960. The carbon to nitrogen ratio started to decrease. The stable isotope N15 became more depleted in the sediments. The concentration of nitrogen increased. This canonical axis #1 sample scores is a measure of the assemblages. So where it had been, there hadn't been much change going on in this lake. And then the composition of diatoms really began to change. And we look at the individual species. one of the diatoms that really became prominent was Asterianella formosa, which is very much of a marker of nutrient enrichment, and shows that this lake was influenced by nitrogen deposition, which reached a threshold level around 1960, stimulating a whole series of assemblages that changed. So with diatoms, we can understand the past and how lakes have changed over time. We can also understand modern sites, streams and rivers. And so these next couple examples are going to be how diatoms are picky about things in rivers, and they're picky about the amount of fine sediment in streams. So if, here's some diatoms growing on a little bit of green algae cladophora. So if there's a fine sediment in this stream, these cells will be buried. You can see that they're fixed, and they actually don't have a way to go anywhere. They're kind of stuck there. And Martin Kelly has made a great illustration of the different life forms. The diatoms that are kind of stuck there, we can call them prostate, or they might be stalked, like Agnanthidium, or arborescent, like a tree. And so as we go in this direction, from Gophanema to Cockanies, the Cockanies is the most resistant to scour and to disturbance, while these are, you can see that they could be easily ripped off. Conversely, the life forms can be better at gathering light as they get up above the canopy, and can get light better than the cells that are below. And then going back the other way is resistance to grazing. You can imagine a caddisfly, a larval caddisfly crawling along the substrate and being able to chow these forms easily, whereas here these can hide in nooks and crannies and are resistant to grazing. So, those diatoms didn't really like to be covered within sediment. Others, like this Gyrosigma, love it. That's the only place where they can live. These diatoms are able to move very well through sediment because they have a well-developed rafe system. That's this line that runs across these cells, which is the way that diatoms move. Some diatoms that can move are going to be able to get away from fine sediment that's deposited on them. That's their place they want to live. So diatoms are picky, and they tell us what their preference is. One of the greats, Dr. Charles Reimer, said, diatoms speak to those who listen. And we try to listen to them and understand what they're saying about where they live. And diatoms can tell us they have particular form and species when they come from a Caline remote river versus an enriched, an agricultural stream that's enriched in nutrients, even to an urban site where both flow, chemistry, sediment have all been highly manipulated. And the species that are present reflect those habitats where they live. So we're working to make this information about diatom pickiness accessible. This is an example from the Diatoms of North America website for the Diatom eucoccines flexula. We show how this species, here's one of these, there are lots of plots here and you can go read about them on site. But this diatom likes to be in fast moving streams. It's resistant to flow, and it does not tolerate human disturbance. Distote is a total human disturbance metric. These are some kinds of habitats and characterizations that we can make about different forms. And this table is really a summary of some of those. I've talked about the life form, the filamentous forms that reach into the water column, and those with attachment. So they may, they cross different categories, whether it's morphology, physiology, behavior, and their ability to gather resources, being nutrients and light, as well as their resistance or resilience to disturbance, and that includes any kind of physiochemical extreme, flows, grazing. So we're working to make this diatom traits accessible. This is a section on diatoms.org, the Diatoms of North America site, where you can find characterizations of each species and what their preferences are. So other solutions is what our group now is working on. We now have a level 1 taxonomic certification exam, and a number of people are now certified in North America as level 1 taxonomists. We are in the process of developing the next level exams, and we have proposed a workshop at the upcoming meeting in Madison, Wisconsin in June on creating a voucher flora, which we expect that this skill in making a voucher flora will be part of the certification process. And you can read more about what a voucher flora is by going to this place on the site. We've known for a long time, one of the problems that people just, I don't know, they call us names and, whoa, how did I do that? They call us names about diatom names, like, you guys, you keep changing the names, and you know, we can't deal with these names that stop changing. We want to make that transparent and easy to deal with. We know that managers get tangled in the messiness of names, and we have a few ways to get through that. One way on the Diatoms of North America site, if you know the old name, you can type in the name that you know, and the first species that comes up is going to be the current name. So we want to allow you to find it, whether you know the original name or a name from 20 years ago or the current name. Even we have some, you know, when people were misidentifying these wrong names in there, you know, like we know people used to call this certain taxon by an incorrect name. We have those tagged so that it's going to take you to the right name. And if you have an entire list of names from a while ago, from one study that was 10 years ago and one study was 5 years ago, and you want to make sure that they're coherent, there's a tool allow you to do that. you can input a list of names, and a function that Ian Bishop wrote using R code will return that list and the USGS biodata taxon name that is current. You can also just input your species abundance metric, your data set, and get back current names. So we want to take the headache out of names. It's something that we can easily deal with scripts and codes, and that people don't need to get stressed out about it anymore. So, and we know that there's a lot of information, and we want to be able to facilitate the use of diatoms. So we want to be here to be available for you, to help you with your diatom data. And let us know the webinars that will be helpful to you, and what's going to be most beneficial for solving the problems that you're facing. So with that, I'd like to acknowledge the generous support of the Environmental Protection Agency, the US Geological Survey, the Institute of Arctic and Alpine Research, that have supported the development of Diatoms of North America, and the Society of Freshwater Science. The certification program that is implemented is set up through SFS, and it's hosted at Stroud. So this has been a really very collaborative effort to try to work through the tools that people need to have to do your work and do your science. And so with that, if we can open it up to your questions and comments and feedback. And I think that the chat doesn't show up on the recording. So if you have a question or comment, put it in the chat box, and Sylvia will read it, and then I or another member of the Taxonomic Certification Committee will try to respond to your feedback.
All right, thank you, Sarah. We have one question from Sherry or Shari, sorry if I'm mispronouncing your name. What are some of the resources that have the various diatoms and preferred environments, information about those?
There are a number of sources in the literature. We don't have those compiled from the literature except on Diatoms of North America. And so we've tried to take the information from those sources and put it there. We also have some compiled metric tables, and I can direct you to places where those metrics are summarized. We hope to be able to automate this process of generating metrics. That's one of the products that we're working on for this year, and to really make it easier to do that.
And then I see in the chat that Jan has added that researchers can contact authors of papers to get species trait information. Maybe Sarah, you could show us on DONA some of the trait information that are included on the species pages.
Okay, that's a great idea. So, if I'm going to first go to get a list of the list of species. So we're almost at 1,000 species right now.
Oh, Sarah, I have a specific request.
Yeah.
Do we have Staurocyrola pinnata on DONA?
I think so. Wait. Okay.
Can we look at the trait information there?
Sure. So we have OT ecology. So here we have size range, motility, its type of attachment, the habitats it lives in, BCG4, and we can show you what that means, and then the distribution, the formal projects that it has been shown in. So BCG4, if we go over, BCG4 are indiscriminate species. So these came from values developed in California rivers. They may be a little bit different in other areas, but they're not going to be a long way off. So what this means is that this taxon, it's not discriminating.
Yeah, I asked you to go to that specific webpage because we have a question from Jiwoo Han. He is a graduate student, or she, I'm sorry, I don't know, graduate student studying paleolimnology and wanted to get your opinion about whether Staurocyrola pinnata is planktonic or benthic.
Oh, what did we say? Because we have it as both benthic and planktonic. Mm-hmm. So this diatom has the ability to attach onto particles, very small. So in that case, it could be attached to sand grains. This diatom can also, because it's very small, can get entrained into the water column, and so it can be part of the plankton. So this diatom is often considered to be mero-planktonic, M-E-R-O planktonic. So it can be can be benthic, but it can also survive in the plankton. So it's not one that's going to be particularly picky about the habitat where it lives. It's able to go across benthic planktonic habitats.
Okay. Another question we have is how often do you anticipate releasing a new webinar? Can you keep me on a mailing list so that I will always know when there is a new one coming up?
Yeah, we are hoping to have them roughly on a monthly basis. And that's a great idea about having a mailing list. Do we have one, Sylvia?
We do not yet. We can create one.
Okay.
Yeah, so please send your email to diatomtcc@gmail.com, and we'll make sure you're on the email list for the future webinars.
Great, thank you.
And you can also follow DONA on Facebook and Twitter for these announcements. All right, another question is from Vania. Hello, I'm working in the Environmental Inspection Agency in Peru. The odd ecology data shown on diatoms.org have used data outside of the US. Are they included in that plot analysis Yeah, so are the data in the plots you showed, are they only from the US?
They are. They are. You know, we have this site is called Diatoms of North America. It might be true that this species occurs outside North America and in Peru, and it might be that it has these same preferences. But we don't know. We're not going to speculate there about what they're doing outside of this area, as we haven't confirmed that they're actually the same species. One thing that is going to be, the size range, the motility, their attachment, their type of habitat, that's the traits are going to carry across from one continent to the next. But some of the, it may be that there are some subtleties in the response to, say, pH, just because there's variance that we wouldn't be able to account for here.
We have a comment from Liz from Queen's University. A mailing list would be awesome. Cool idea for a webinar. We use the Diatoms of North America website to confirm our taxonomy and keep up with new names.
Great.
And just in case you can't see the chat box, that email I said before about signing up for the email list is diatomtcc@gmail.com. And we're getting some suggestions about webinar topics. We will send you the link. You can go to the link on Sarah's slide.
Here,
yes, there it is.
Is this the whole, that's a long link, oops.
There is a short link.
A short link.
Mm-hmm. On your slide.
Okay, let's see.
So this is where you can tell us all the different topics that you are interested in learning about. You can rank the list that we created and tell us if they are of high interest, medium, or low interest, so we can determine which webinars to focus on. And there is space at the bottom of the form to input your own ideas if we didn't think of them.
Don't see a topic you want to learn about? Tell us, let us know.
Okay. I will try to copy and paste the link into the chat as requested. So once the recording is ready, we will include the link to the topic survey, this form you're seeing right now. And you will get a link to the YouTube video of the recorded webinar. And you will get the slides as a PDF. Oh, thank you, David. There we go. The link is in the chat.
Great. So you should be able to copy and paste from there.
Just a comment from Luca. Hi, Luca. He says, thanks, recording is great. He could only tune in for the last part.
can catch the beginning when we post it.
Hi, Sylvia. I'm sorry. And yeah, I'm looking forward to learning more as usual. And you're doing an amazing job, you all.
Thank you, Luca.
Thanks.
Hopefully we'll get some more Diatom of the Month posts from some of the attendees. I guess we'll send emails around as well, maybe.
Yeah, tell us about the Diatom of the Month.
Oh, what can I tell you? We got beyond 50 posts, and it's tough to keep it going, especially with the different research focus for me now and plastic pollution. But Xavier and others, especially Nick, lately have stepped in a lot. So yeah, any additional feedback will be appreciated as well. We can probably write 10 posts on your work, this group's work.
So who's in charge of Diatom of the Month? Which group?
Well, Xavier and Annika Vilmi, Nicholas Schulte, and yourself and myself, with a bit of time that we have. But yeah, it would be incredible to keep it going for another year and then maybe another one, and get some senior authors to give ideas for topics or write themselves as well.
We could keep it going forever.
Yeah, it's kind of
there are many diatom species.
Yeah, it's kind of scary though.
And just so everyone knows, I think I can leak this information that the International Society for Diatom Research, we are launching a new website. It should be about March 1st, and that's where Diatom of the Month will be hosted.
Yes, so thanks for saying that.
Yeah, so it's going to be moving over to that location, and I know you guys are working on making that happen and all the work that goes into it, so thank you.
Well, thanks, Xavier, and yeah, I'm less connected than I would like to be, but maybe, if the next job is more Diatom related, that's gonna be easier for me.
But this is a great audience, people who are interested in diatoms.
Oh, yes, indeed.
You would like to make a post on Diatom of the Month, please contact us.
And simplify the language if you can, because we are trying to reach non-specialists as well, if we can.
Okay.
So from papers to blog, that's what I'm talking about. Yeah.
Oh, we have a message. A future attendee of the Ecology and Systematics of Diatoms course at Iowa Lakeside Lab.
Great. Okay, anything else? Anything from our other Diatom Taxonomic Certification Committee folks?
Josh Cooper says he looks forward to topics posted in keeping our community of scientists, researchers, managers growing.
Thanks, Josh. Great to hear from you.
Okay, I don't see any more questions. Oh, one more from Gina LaLiberty. Will there be a certification exam at ASLO SFS in June 2020 or just a voucher workshop?
I think we're holding an exam, aren't we?
Yes. So we will have both things, certification.
Yes,
exam
both a certification and a workshop.
Yes.
So Gina, you better be there. We're expecting you.
All right. Okay, one question here. We still have a few minutes. So the question is, are the benefits of studying diatoms, what are the benefits of studying diatoms as opposed to taking direct measurements of pH, alkalinity, temperature, etc.?
I'm just laughing at you. And that's funny. Okay, that's a great question. So with instruments that can be out continuously recording, that's a great thing because you can measure those things continuously. Diatoms, you know, we can't always do that, and so diatoms are able to tell us what has the pH been like in this lake. You know, was there a release of material overnight or in the past? So it allows being an integrated record of a site.
Yes, so if you're specifically wanting to know what is the biological response to these changing conditions in pH, alkalinity, temperature, then diatoms are very useful in that use. It integrates what conditions are happening where those diatoms are living. So, and if you take a direct measurement of the pH, you know the pH of that particular time and place of that particular water sample. So you may not be seeing the broader picture of what's going on in that area.
I'd also add that, you know, it's really, we end up caring about pH or alkalinity or temperature really when it affects organisms. It's really our marker of, is this beneficial for life or not? And so much of the language is towards protecting life. And so just having a pH measurement doesn't really tell us about what is the condition for life. We want to know the health of the organisms that are there in addition, or maybe in a different way than saying, can they tell us about what the physical conditions are? Another question is, what is their health? Are they in good health, or are they altered in some way from what's a natural condition?
We have a question from Gustavo. The main objective of the webinar series, will it be to support managers and non-taxonomists, or does the team intend to bring topics for specialists at different levels too?
Yes, we'd like to address that wide range of audience. It may be that a particular webinar is directed at managers, and another one might be directed at specialists. Like Sylvia could tell us about the genus Mastigloia in North America, its diversity, distribution, ecology, problems that analysts might come across in trying to make the identifications. So we would like to have a spread of those talks, but it really depends on all of you letting us know what it is that you want to hear, and being able to you can recommend particular specialists that you would like to hear from, and we can try to get them to talk about their work. I think that would be really powerful to have this means for us to continue to connect with each other during the times when we're not at professional meetings or you know, in between, to really make this part of a you know, community of practice, that we have our closer connection with colleagues.
All right. And if you really want to dive very deeply in learning about diatoms, Mark Edlund, my co-instructor, has posted a link on how to get information on the Iowa Lakeside Lab Ecology and Systematics of Diatoms class. It's 4 weeks of fun times at a field station, learning how to collect and identify diatoms. You know, this webinar series will cover a lot of topics that are important to many different aspects of diatom research. But if you want to get the in-person experience, having training in diatom research at the microscope with other diatom researchers, this is a great place to get that training and experience.
So Gustavo says, thank you and congratulations for the initiative. And thanks to you. We appreciate people joining us and taking part. And yeah, we depend on your feedback and ideas for this to be successful. So we'll try to do our part to get a mailing list and posting information online for people that weren't able to join us in person. And we'll have a posting on Diatoms of North America, Facebook, Twitter, so that we can stay connected. So if there are no more questions, I'll just thank everyone for coming. And we look forward to your feedback and offering the next series. We hope to have this series of webinars once a month. And Sylvia has posted the link. So you can, Diatoms of North America is at diatoms.org. On Facebook and Twitter is at Diatomologica. And you can contact us by email at diatomtcc@gmail.com.
All right. Thank you, everyone. Thank you, Sarah.
Thanks, Sylvia. Thanks, TCC. Thanks, everyone, for joining us. And we hope to hear from you, and we hope to hear, to get into some specialized talks. I think that would be great.
All right. See you next time.
See you next time.
Thank you.
Bye.